Okay, so I think this is a very exciting time in neuroscience because there's so much experimental data. And there's experimental data at many different scales of the brain. And the amount that is generated every year is pretty impressive. So we've seen a lot of that today. And this is another example that I wanted to show, some of the data sets that we are starting to use right now. And this is electron microscopy. It's 1 millimeter cubed of the mouse cortex. And it includes all the 200,000 neurons inside this volume, as well as 500 million synapses. So the level of resolution of this data set is pretty impressive. So you go down to the level of single synapses. So you can basically reconstruct this full millimeter of cortex in mouse with all the neurons and all the synaptic connections. There's also a similar data set for humans that we're also working with. But of course, there's a lot of data at different scales, as I mentioned. So we'll have a lot of genetic, molecular level data that we've seen today as well. We have a lot of electrophysiological data. Even the previous data set that I showed also comes with calcium imaging. So you can know the actual response of each of these neurons that we are seeing anatomically. We can see the functional response as well. And so the question is how to interpret and integrate all these different experimental data at different scales using different techniques. How can we integrate it and make sense of it? So one useful tool, we think, is computational models, and particularly the type of models that we build, which are multi-scale models where we integrate data from all these different levels. And so, of course, the idea here is that once we build the model, we have to validate it. So we have to make sure that the model is actually behaving as we expect it to do. So we do that by showing that the model reproduces the responses measured from animals in vitro and in vivo. And then once the model is validated, we can, of course, use it to make hypotheses and predictions that can be tested experimentally, and even potentially evaluate treatments for different disease, and then also try that in real life. So in the little time we have, I'm going to try to show one example of that we've done with a model of the primary motor cortex circuits in the mouse. So this is a model essentially of a cortical column in the mouse. And this is based on primarily in data from a single lab, from the Gordon Shepherd Jr.'s lab, who had data at many different scales here. So this model includes 10,000 neurons and 30 million synaptic connections. The neurons include 8 different cell types, including excitatory and inhibitory cell types. Each cell type reproduces the morphology and also the physiology of real neurons. So they are mapped by modifying the ionic channels of the different neurons to reproduce the firing rates of each one and their different conditions. And of course, we also included a lot of information at the synaptic connectivity level. So we have like 4 different types of synapses with the real parameters. We have connectivity at the local microcircuit level obtained from optogenetic studies. We have connectivity even at the dendritic level. So we know the distribution of synapses that come from different types of neurons in the circuit and how they are laid out in the dendrite. And we have information about the long-range connectivity, so different inputs from different parts of cortex and thalamus and where they project into this circuit. So we basically put all this together, this information from over 30 different publications, and then we started evaluating and comparing against in vivo data. So for this, we started a collaboration with another lab who does in vivo studies in mice, M1. And they have a nice setup where the rat is running on top of a treadmill, and they do whole cell recordings of the rat neurons in mouse M1. So you can actually get information about specific cell types while the mouse is performing behaviors. So what we did is, you can see here, this is a raster plot of the activity. You can see the raster plot here. So the y-axis is the 10,000 different neurons, and the x-axis is 2 seconds of simulation time. And we simulated here 2 different behaviors: quiet when the mouse is not moving, and movement when it's in the treadmill doing locomotion. And so we show that we can reproduce the firing rates, the activity of different cell types in the circuit. So I'm just showing here a couple of examples, but we have actually matched statistically the responses of neurons in the 6 layers and also different cell types in each layer, and both for during quiet and also during the movement state. So it's behavior dependent responses and also cell type specific responses. Similarly, with the model, we can also simulate these other measures, like local field potentials, for example. So we place a simulated electrode in our simulated cortical column, and we generate these LFP signals, also related to different behaviors, to quiet and movement. And so here we show the experimental data from the in vivo study, and also the model. And we also compare statistically how the responses change from one behavioral state to the other, and we show that the model reproduces these changes in the oscillatory activity in the LFPs. So, for example, when you change from quiet to movement, the delta oscillations, the power of delta oscillations is reduced, and the power of gamma oscillation was significantly increased. And we see here how the model matches these changes in the LFP. So once we had done this pretty thorough validation of the model, we started using it to generate some hypotheses and predictions. So the in vivo study showed that there's 2 requirements for the mouse to move, and one of those is inputs from the motor thalamus, and the other is noradrenergic inputs from the locus coeruleus. So through using the model, we were able to analyze these 2 inputs in a very systematic way, so providing different values. And you see here in this plot, in the y-axis is noradrenaline. Sorry, in the y-axis is the motor thalamus, and the x-axis is noradrenaline. And we can systematically evaluate the expected response, in this case of the layer 5 neurons, to different levels of these inputs. So these are specific predictions that can then be tested by our collaborators. And of course, the model can go beyond that and provide predictions for specific cell types. So within layer 5, there's 2 major cell types, the ITs that project intracortically, and the PT cells that project down to spinal cord. And so the model predicted an interesting thing, which was an inverse response between these 2 cell types. And so our hypothesis that came out of the model is that these 2 cell types were interacting through PV neurons, paravalvumin interneurons, and there was mutual desynaptic inhibition. So we actually found some data to support this already, and some of the collaborators are already planning studies to try this hypothesis that came out of the model. And so that's nice because we're essentially closing the loop. We started with the data, the models provide some predictions, and then the experimentalists can test them and again fill data for the model.
1 minute. I know you're talking for 2, but...
Okay. So another prediction from the model was this. The experimental data showed that if you cut the connections from locus coeruleus, there's a change in behavior, and the mouse doesn't actually perform the behavior correctly. So with the model, we were actually able to come up with a hypothesis at multiple scales of why this was happening. So the neurons, these PT neurons in layer 5, when you reduce the noradrenergic inputs, the HCN channel increases the conductance, and that reduces the integration of synaptic inputs, which in turn increases the firing of these PT cells. So this was essentially linking the molecular, the cellular, and the circuit level to the behavior correlates that we were seeing. So since I have a minute, I'm going to go very quickly over this. The models can also be used, for example, to elucidate where different oscillations are coming from, what specific cell types and populations are generating theta oscillation, for example. And this, of course, can help potentially treat disease like epilepsy, schizophrenia, Parkinson's, which are related to oscillations. And finally, a quick mention So we developed this software tool that helps build, simulate, and analyze these kind of models I'm describing at multiple scales. This is funded by the NIH, and it's supposed to help people who don't have too much experience programming to build these models more easily. And we've been lucky that the tool has been well received in the community. So there's over 40 labs that have used the tool already to build models of many different aspects of the brain, including many diseases that you see here: schizophrenia, ischemia, epilepsy, depression, Parkinson's. Different stimulation protocols as well, things like TMS, optogenetics, the effects of ketamine. We can also simulate things like EEG and MEG, even now fMRI. And we have models of many different parts of the brain, lots of cortical regions, thalamus, striatum, amygdala, hippocampus, spinal cord, and even some models of the cardiac circuits. So thank you, everyone.
Thank you.